I'm very, very excited and pleased to introduce our keynote speakers this morning, colleagues who have become friends over time. My hope is that their story enlivens and focuses your attention, that it transforms your outlook from very abstract and theoretical and scientific to a more personal and lived experience. It is my pleasure to invite to the stage Ms. Linda Lafontaine and Dr. Jason Karamchandani. So Linda Lafontaine, our dear friend Linda, is a patient diagnosed with hereditary spastic paraplegia, HSP, an advocate for the HSP community, and a self-described patient on a mission. And you'll feel the energy and the passion that Linda can share. You'll understand why she's such a great patient partner for us. My dear colleague and friend Jason here, Jason K., associate professor in the Department of Pathology at McGill, neuropathologist with us at the Neuro, the president of the Canadian Association of Pathologists, and most importantly for this talk today, the director of the Neuro's Open Science Repository, CBIG, which stands for Clinical, Biospecimen, Imaging, and Genetics Repository. Please join me in welcoming Linda and Jason to the stage.
I start. Hi, everyone. Well, thank you to Annabel for that wonderful introduction. Thank you to Conscience for inviting me, to my partner in crime, Dr. Karamchandani. And thank you to everyone for listening to me. You don't really have a choice. But seriously, thank you for giving patients a voice. That's very, very important. And shout out to the Spastic Paraplegia Foundation. They're an American foundation, but I'm here because of my persistence, we'll call it persistence, and because of my massive patient network, and it all started with them. I'm pointing there because it's being recorded for them. No matter what happens in the States, it's very entertaining. I love them. They're part of my HSP patient family. That will never change. All right, so I'm going to tell you a little bit about hereditary spastic paraplegia. Remember, I'm very wobbly. Okay, but I'm a patient. I'm not a biologist. I'm going to tell you a little bit about it, but I'm not going to go into very much detail. So I've been living with this condition for just a little over 20 years. As you can tell, I named my wheelchair Wilma, and she's pink. It's a she because she's pink. So I fully understand how serious my disability is, my disease is, but I've been hearing it for 20 years, so I don't care. It's a rare neurological condition. It's a bunch of little conditions all rolled into one. There's a bunch of different pathways. My pathway is genetic, and the mutation is on my SPG7 gene. My mutation is pure and uncomplicated, so it's pretty straightforward for HSP. It's progressive, which means it gets worse over time, slowly over time. What you see today might not be what you see in the future. In 5 years from now, I don't know, it's a surprise. We can find out together. But patients are constantly living with uncertainty. We don't know what's going to happen next. There's very early onset of this disease. There's very late onset. So babies can be affected, toddlers, teens, adults, seniors. Doesn't matter. It affects everybody. There's the stuff you see in me. So why I'm in a wheelchair, there's massive muscle weaknesses and spasticity. So my muscles are always on. There's the stuff you don't see, the bladder issues. I have kidney issues, circulation stuff going on, inflammation, pretty much all of my joints. My hormones are all out of whack, which affects my digestion and my sleep and a whole bunch of really fun party tricks. And then there's the stuff you don't see in me. There's the cognitive issues, the stuff with the voice or the swallowing. There's all kinds of nerve issues going on with this disease. So some people, they have a problem with their vision, with their eyeballs. My vision sucks, but it's not HSP related. They have all kinds of, and the pain. The pain is par for the course for some people with HSP. There's one of my friends was walking around with a broken bone, but she's got pain all the time. So the pain of the broken bone is just par for the course. My journey, my personal journey to HSP, we don't know a whole lot about this disease, so it's very often misdiagnosed. It's mistaken for other stuff. For me, it was mistaken, but not for another disease. It was mistaken for pregnancy. I was pregnant in 2001. I was pregnant again in 2002. Somewhere in there, the back pain and the weird stuff started to happen, but we all thought it was because of the little human I was drawing. And then after that, I gave birth July 2022, not 2022, 2002. Then it was postpartum. We thought maybe the funky stuff was postpartum. Somewhere in 2003, I went from OBGYN and family doctor to neurology. I don't know exactly. I don't remember how the leap happened. My family doctor doesn't remember, and it's not written in my file. So we're going to start with 2004. I was clearly in neurology at that point. My journey to a diagnosis wasn't done. No. We had all kinds of tests from 2004 to 2007 just to eliminate the low-lying fruit. And then after that, in 2007, it culminated into an MRI, which confirmed the HSP diagnosis. I didn't have the SPG7 diagnosis then. We still had tests, 2 more MRIs, genetic testing in 2012, I think. Maybe it was 2013, I don't know. Doesn't really matter. Then I got the SPG7. But through all of that, nothing has changed. Even today, nothing has changed. The patients are still living with uncertainty, with the fear of what this disease is going to deliver to them tomorrow. What fresh hell HE double hockey sticks is going to happen next. They're still living in grief for what they could do before that they can't do now. So that's my overall motivation for Patient on Mission. For doing everything that I'm doing, my motivation is to advance science, meaningfully and through open science. I want to define meaningfully. I want to advance science so that it's advantageous for people who do science to make discoveries. That's why I'm doing everything through open science. With that, I will turn it over to my partner in crime, Jason Karamchandani. He's the associate director. God, I can't think.
I'm a pathologist. That's what I am.
Oh, there you go. Yes, he's also a rock star. Yeah? Yeah. He's the scientific director at the CBIG, the Neural Institute.
So I think Annabel already put this very eloquently.
Thank you.
We're going to go back and forth.
Yes.
So Annabel already described the very significant need that we see every day at the Montreal Neurological Hospital. And I'm not here to make excuses, but there's a reason that there has been disparities in progress in the different areas of healthcare. So this is a quote from one of the many tomes of Hippocrates. So more than thousands of years ago, there were multiple books written about how to take care of patients. And essentially what he's saying here is if you see the skull or the dura, walk away. You will not be able to help that patient. And the reality was that was true for most of human history. In fact, it's just about 100 years ago that we finally had surgeons who were able to operate on the central nervous system and its coverings without killing the patient. These were two of the most preeminent about 100 years ago, and I'm going to focus on Penfield because there's a street named after him just up the hill, and he founded the institute that I'm so proud to be a part of. Now, one of the themes about today is patient partnership, and I'm hoping to stress to you that the history of the neuro embodies this concept of patient partnership. When you walk through the door of that hospital in the 30s, we had not even mapped out the eloquent areas of the human brain. You were going there for clinical care, but at that point, we didn't even understand the consequences of surgical intervention. While doing these operations and with patients who were awake, Dr. Penfield mapped out the human brain and produced this image which every physician on the planet knows, the homunculus. And it showed what elements of the brain were responsible for which areas and domains. And you'll see here that's from 1948, and with millions of dollars and thousands of MRIs, it was updated just about 2 years ago, and I would describe those as fairly minor revisions. So Penfield did an amazing job. This is a real slide from a real patient, a young boy who presented to an emergency room in Texas. This image was provided to me by my first chief resident, Dr. Chandra Krishnan. This patient presented with fatigue and bruising, arrived in the emergency room, had a blood draw, and within 45 minutes, a diagnosis of acute leukemia was made. But it's even cooler than that. So inside this cell over here, you'll see this red rod, this little linear structure. That actually allows us to subtype this leukemia. This is acute promyelocytic leukemia. And in fact, that dictates a very specific treatment. So within 45 minutes of getting to the emergency room, this patient already had a precise molecular therapy for his pathology. I think that's fantastic. And in some small areas, we're making progress in the CNS as well. This is another young boy, just a little bit older, and what you see here is a biopsy of a cervical medullary brainstem lesion. And that's not a resectable tumor. Obviously, the brainstem is very valuable real estate, and if you permit me to whinge for a second, this is one of the challenges of modern medicine, is that we have to do more and more with less. This is our specimen. That's it. This, by the way, is a millimeter, 1 millimeter. So we've got almost nothing. And it's generally a bad thing when a pathologist can count all of the cells in a biopsy, but we understand that this is an important area. And in fact, we can demonstrate that with immunohistochemistry. So every one of these black wires you see in this picture is an axon. Those are the cables that bring information back and forth from the central nervous system to this patient's extremities and peripheries. So there is no bite, there's no biopsy without morbidity here. And this is just one of the many markers we do in pathology which shows that these cells are dividing, meaning if there is no intervention, this expansile mass lesion will eventually kill the patient. But we've made some progress. This is actually from 2015, and we have an antibody against a specific mutation, BRAF V600E. This is accompanied by a precise molecular therapy, which worked. So you start with the tumor on the left is at presentation. The tumor on the right, which went from 11 centimeters down to about 2.5 centimeters after 6 months of therapy. Not only did this work, and not only did this patient have a durable response, this therapy has evolved since then. So in the last 10 years, not only do we have a new BRAF inhibitor with slightly easier side effects, we've added on MEK inhibitors, and they have superb clinical responses. So we're making some progress. This is good. But then we compare it with other neurological diseases, like Parkinson's. Just like in leukemia, in 1950, you walk in the door and everyone died. Within 5 decades, we were able to treat the majority of those patients, and today we treat nearly 9 out of 10 with a durable 5-year response, and that's only getting better with the new cellular-based therapies. Parkinson had its first major therapeutic innovation also in the 50s and 60s with L-dopamine. And really, there's been nothing fundamentally transformative since that was introduced. And that's the case, unfortunately, for HSP as well. So if you look up modern therapies for HSP, we see a list of repurposed drugs, some to do central nervous system relaxation. We see benzodiazepines, muscle relaxants, but there's nothing here that gets at the heart of the disease. We are treating symptoms, not the underlying pathology. So why is this disparity? One of the explanations is that in leukemia, you have ready access to the cells that drive the disease. You can put them in a dish, you can poke them, you can perturb them, you can add reagents and see how the cells respond and react. That's where we make good progress, where we can understand the biology that drives the disease. We don't do brainstem biopsies to look for dopaminergics in patients with Parkinson's disease. So we don't have access to the cells that really help us understand what is going on. Now, there have been plenty of successes in rare diseases, largely in children's cancers. So every Canadian in this room knows the name Terry Fox. He died from osteosarcoma. Now, in 2025, about 70 to 80% of osteosarcomas are treatable, even if they present with pulmonary metastasis, which is ultimately what killed Terry Fox. We are making progress by aggregating cases, and there a sense of attention and critical mass is helpful. So if you have enough people looking at a particular disease or problem, we tend to make good progress. So with that in mind, in 2014, the director of the Montreal Neurological Institute decided that we needed an open science biobank to try to share materials and cells to help advance our understanding of neurologic disease. And I'll be going into detail about that later this morning, but I do want to emphasize that all of this is possible because of the donations of patients. Everything that is in this collection comes from patients. And our philosophy is very much that putting things in a freezer doesn't help anyone. Collecting data doesn't help anyone. It's only when you get those cells into the hands of scientists or data into the hands of investigators that you're actually doing anything. And that's what drives the philosophy of our open science repository. Now, I put these slides together before I knew Annabel was going to be doing the introduction, so I have to absolutely thank the Tannenbaum Open Science Institute. Everything we're going to talk about today is a consequence of an original aspirational investment by the Tannenbaum Open Science Institute, which was strengthened by a significant contribution from Brain Canada with a project led by Dr. Boulot. So the Neuros Open Biobank, we affectionately call it CBIG. As Annabel mentioned, that's clinical biospecimens imaging and genetics. That's a multimodal repository containing longitudinal, deeply phenotyped patients that we follow through time. I'm going to skip over this because you don't need to go into too much detail, but I do want to help everyone understand that for the WHO, it's basically everyone. It's pediatric patients, adult patients, people who require substitute decision makers. Our first version of this in 2016 did not touch pediatric patients. For those of you who do human research, if you add on patients under 18, your paperwork gets about 5 times longer. But the community came back to us and said, you have to be able to engage in this population, because when do you diagnose autism? It's not when patients are 19 years old, it's when they're children. And so we went back to the drawing board just as the project got started. We collect a wide range of stuff and data. And I think this is really interesting because this is where Quebec has managed to do something I think extremely well. All RAMQ cardholders in this province have a Dossier du Santé du Québec. Any scan that was acquired here in Quebec, we can automatically de-identify it, strip it of identifying information, and push it into the research space. In our MS cohort, that's more than 900 patients containing thousands of scans. Those are scans that we as the public helped pay for, And now, thanks to this platform, we can actually reuse this data. We can use it for secondary use of this data for research and investigation. We collect data from things like chart review. We have automated data import from structured data in our medical information system. There's a lot of different ways we collect this. So this was the original vision in 2016. You take patients, we take some materials for them. We share them with my excellent colleagues in the early drug discovery unit, and I see Dr. Durkin sitting over here, who takes these cells, reprograms them, and turns them into iPSC cell lines. We can modify them, but we now have the cells we need to study human neurologic disease. And it works. I jumped through the roof when I saw this all those years ago in 2017-18. This is a midbrain organoid. The reason this blob is brown is that those cells are producing neuromelanin. Those are the dopaminergic neurons that are affected in Parkinson's disease. And this is a patient-derived organoid. This is a new tool we have to study these complex neurologic diseases. So here's where we are in 2025. We have more than 80,000 specimens from over 4,600 patients. Our cohorts reflect the patients that walk through the front door. That means we're going to see lots of patients with Parkinson's, with ALS, with rare neurological disease, with multiple sclerosis and neuromuscular disease. And most of these samples have been sequenced, and I have some good news to share with you later today as well. When I started this project, I thought as a pathologist this would be about collecting materials and freezing them and sharing them, but it turns out I was wrong. This is actually an informatics project, because if you don't have an efficient way of sharing what you have, your collection with people, and there's no way for them to easily get their hands on, download, and analyze the data, you might as well not be doing anything. So luckily, we had an initiative that was started more than 20 years ago. Again, we use lots of acronyms in the neuro, so don't worry about the names, but LORIS was the longitudinal online research imaging software that was designed for phenotype and imaging, longitudinal imaging. So we took it, modified it, we added on an integrated laboratory information management system so that all of the information about any of the specimens that we collect is available to anyone who would like to investigate what we have in the collection. And the major initiative the last several years was to integrate the bioinformatic data together with Dr. Sally Farhand, who's the director of the Neuro's Bioinformatics Core. This is just a nice example of the publicly available iPSC cell line catalog, again, done together with the Early Drug Discovery Unit. And this allows people to filter. In this case, I've already filtered by disease, but we have numbers of different diseases. You can filter by gene. You can even filter by whether or not the cells have been reprogrammed or not. And I think it's really interesting that, as I understand it, CRISPR patents are now starting to be enforced for industry partners. And so you can imagine that this is very relevant to some of the partners working with these materials. I'm extremely excited to be able to tell you guys about this. This is fairly recent news, but the largest grant in the history of the University of Ottawa was awarded several months ago. It's the Brain Heart Interconnectome, and it's a $109 million CEFREF. This organization has opted to use the CBIG platform for the storage and sharing of all of their data. So now we have 2 universities across 2 different provinces, avoiding duplication, not wasting time starting from the ground, starting from scratch, but leveraging each other's wins, and the ecosystem itself will improve as we bump into real-life use cases, solve problems, and we're going to get stronger together. And I think that's extremely exciting. I want to make sure we get back to Linda, so I'm going to go fairly quickly over this, but there's all kinds of bioinformatic data. There's whole genome, exome, RNA-seq, single-cell RNA-seq, and all of this is being integrated in the database and will be made publicly available this summer. We also share this data in multiple ways. So the CBIG data portal is one way to get your hands on this. You can also go to the Canadian Open Neuroscience platform and find packaged versions of these data sets, which are obtainable via controlled access. And I think we'll go back to Linda over here.
Okay. Yeah, so you can see what we're going to do today. In real life, we work together. So we're going to try and repeat that on stage. I talk a little bit, then he talks a little bit, I talk a little bit, back to him. But I started this thing. I'm going to finish this thing. Okay. All right, so I helped lead a cohort of HSP patients housed in the Neuro Open Biobank, which you just saw there. I use clinical information from my patient friends, my friends and colleagues. I invite them to participate in CBIG. CBIG then collects their clinical information, transforms this information into raw data, and that data fuels research, like you just heard. And it's that research, the open science platform that enables me to do what I do. So what I do is I get information, I use the information, I make connections. Before I go into the connections, I want to share with you how I found CBIG. So again, I'm a patient, I'm not a historian. I'm going to take you back to 2021. Going to be very quick. Became an ambassador for the Spastic Paraplegia Foundation. Again, I'm pointing at the iPad that's filming me right now. Ambassador is just a fancy word, means communicator, sender of emails. They're an American foundation with some Canadian members. It's just communicating between an American foundation and its Canadian members. Canada being a huge country, I had some members in Quebec, so I always speak in French and in English. I met a woman in Quebec, and she told me about her research. She had found the neuro and Dr. Rouleau. I'd never heard of the Dr. Rouleau and the neuro, so I Googled him. Turns out Dr. Rouleau is legend. And the neuro, they're in Montreal, they're in my backyard. So I took my patient on mission south and on a train, and I went to meet with Dr. Rouleau face to face. He's a fabulous man, and he just casually introduced me to Dr. Karamchandani, my partner in crime. This was end of 2022. See, already skipped a year. I remember sitting down where Dr. Karamchandani told me, he introduced himself, told me how CB works and all that, and I kept thinking, I don't remember when, but I kept thinking, holy jumping, that's what I'm looking for. I was so excited. At the end of 2022, I personally contributed to CBIG. I was very excited about that. I'm glad I did it because what I can tell you now, open science, because it permits patients to participate actively in research, open science empowers patients. And I can also, because I've done it, I can explain how open science empowers patients. It's very simple, it's no-brainer, well, for me. Open science, it gives the patient control into their health that the disease took away. That's what it does. That's how it empowers patients. So in 2023, after I had calmed down from finding Rockstar there, I just took a flyer saying, hey, I know my patient friends feel like I do. They want to advance science. They want to, get research going. I sent them an email and asked them to do what I did in 2022. It worked. It worked. It took a flyer, sent an email. It worked. And a small cohort was born in 2023. At some point, we added on some PLS patients to that. Now the training wheels are off. I'm expanding the cohort. And it all started with an email. So with that, I'll turn it back to Dr. Karamchandani.
So I think that's really interesting. We're starting to touch on some of the ethical and governance aspects of this, which I won't spend too much time on, but I think are remarkably important. In the early days of this project, we didn't have a large team, and I participated in the direct consent of hundreds of patients. The question that I received most frequently after explaining open science was, isn't all science like this? There is an assumption from our patient partners, people who contribute to these projects, that scientists share data openly, that the scientific community is barrier-free. That's a misconception, as we all know, and I'm very happy to see the momentum pushing things towards what patients think is taking place right now. And open science, open means different things to different people. In the world of healthcare information, obviously it's not putting it all where somebody can find everything on Google. So we have tiered access. We have open metadata, things that cannot harm a patient. As soon as you get down to the individual patient through time, that requires registered access. Essentially, you agree to the terms and conditions you would, like when you download an app on your phone. It happened to be drafted by a very talented lawyer who kicked this conference off this morning. And so it's just essentially a sense of community standards. And then finally, for things like genetic information or actual biospecimens, where in theory you can take it to your lab and sequence the genome, that goes through controlled access, and there's actually an open materials and data transfer agreement that we execute through McGill. All of the documents that we use are publicly available. So the framework for CBIG is published openly. All of the SOPs for every sample we obtain is published openly. Our community standards is downloadable right off the web. We try to share as much as we can for anyone that's looking to emulate this model. All right, so we're going to talk a little bit about the HSP cohort, which has been a really interesting 2-year journey now. And this started because of Linda. So Linda said, I want you guys to use this operation to focus on HSP. And we said, sure, why not? So as Linda points out, Linda has this incredible network, this community of patients with HSP, which is a rare disease. And she has colleagues and friends across the country and even around the world. And so what we did is Linda will approach her colleagues, inform them about the nature of the project. If they choose to consent, they go through the official and legal consenting process. We send out kits to patients across the country to collect DNA samples. We go ahead and sequence those samples. And what I think is really gratifying and rewarding is that where possible, we return those results back to the patients themselves. So it's not an abstract altruistic, it is always an altruistic donation, but where we can help, we are obligated to do so, or I feel like we're obligated to do so. So once they join CBIG, then they sort of go behind the little dotted line, little firewall, and then we're no longer able to share that granular information with Linda, but she has a global sense of the cohort. So last year, we had a public outreach educational event. So for people that were in CBIG, for people who had not yet consented to CBIG, we did an informational session that went around what to expect from genetic testing. Because we use words and we have a vocabulary that doesn't necessarily mean anything to patients. So what we did here is we essentially prepared them for 3 outcomes. You are going to have, we all have lots of variants that mean nothing, so we're going to have benign variants. We have the dreaded VUS, the variant of unknown significant, meaning we don't know what the impact of that genetic alteration is. And then we have likely pathogenic variants. So we worked with an organization called CAN-HSP, which is ironically also led by Dr. Rouleau and Dr. Ganor, both based at the Neuro. And we interrogated the genomes of patients that had never had genetic testing. And we were able to come up with a diagnosis, a molecular diagnosis, in just under half of that group. So we had pathogenic variants in just under half, a likely cause of the disease in one patient, and then a bunch of patients where we were still unable to identify a cause of the diagnosis. And even though that's frustrating, that last half, that's where the real action is, because just because the test is negative doesn't mean they have HSP. It means that there is another cause of HSP that we haven't figured out yet. That's really where the research and the clinical come together in a single space. And I think it's really timely to talk about this project because 2 weeks ago we were, thanks to the work of Dr. Ganor, again the leader of the CAN-HSP project, the Institute and Dr. Ganor have received a significant amount of funding from Genome Canada. What that means is all of the samples in CBIG from patients with characterized neurological diseases or uncharacterized rare neurological diseases are going to have their samples sequenced. And so the impact that we were able to deliver to the HSP cohort will now be shared with a much larger cross-section of those patients that participated in this project. And particularly for rare neurological disease, Dr. Tetreault on the right here is doing the long-read sequencing, which is particularly good for some of the nucleotide expansion pathologies and other rare diseases. So this is sort of where CBIG is today. And this was entirely possible because TOSI funded this before everything got off the ground. But as you can see here, this is now the hub for several CHR-funded networks, several pan-Canadian disease networks, including the Canadian Open Neuroscience, Canadian Open Parkinson's Network, the CAPTURE project, which is ALS, CAN-HSP, and has since been part of several large national grants, including the Genome Canada grant I just described. And we're having interesting collaborations with the University of Ottawa and a brooding new collaboration, which I think would be very cool, with the University of Western Ontario. One of the ways we try to measure if this is working or not is whether or not what is this doing? What comes out of this? And we've participated in over 100 and 105 scientific collaborations with academic and industry partners essentially around the world. So we've dealt with multiple countries, both academic in the central middle column here and in industry on the far right. But we've worked with most of the provinces, many of the universities, and both small and large industry partners. And finally, all of these collaborations are conducted in the open. So what that means is, if there is scientifically valuable data generated from our partners, after a mutually agreed upon embargo period, that data is returned to CBIG and made available to subsequent investigators. And so I've got two examples from industry partners, and this one is about the level of alpha-synuclein antibodies, and perhaps more relevant or tangible is a collaboration with Takeda, in which they did RNA-seq on 50 samples from patients with ALS. At the time, that cost around $3,000. That's $150,000 of investment that can then be leveraged by the wider scientific community. So I'm finally going to close here with the fact that this was designed to also facilitate clinical trials. And so we now have cohorts of genetically characterized patients that are ready to go for clinical trials. This is an example from 2021 with ALS being a rare disease. A rare cause of ALS is ATXN repeat expansion. We identified 4 patients, and we even identified one that made it to the clinical trial. And I'm just going to close by saying I'm familiar with some of the work, the incredible work being done by M4K, and that's the final leg. It's wonderful to do all these things in the open, but until we produce meaningful therapies, we haven't delivered to the patient community with our shared goal of accelerating the development of treatments. But I think that the work being done in M4K in a rare neurologic disease for diffuse midline glioma is an incredibly exciting model, and it's a fantastic group. So I'm going to wrap it up. I see we're at the end of time, and I'll thank all of you guys for your time this morning. Linda, back to you.
Okay, thank you. All right, so I take what you just heard, and I try and make meaningful connections for patients. I use everything I do it through open science, and I make connections just because open science permits me to do it. So I do it just because I can. Nice words, but how do I do that? Okay, well, on December 5th, I brought SPF doctors, the Spastic Paraplegia Foundation. I brought them to CBIG. As I was writing my speech, I realized they're not SPF doctors. They're neurologists. It's not what you would see on their signature block. Their CVs wouldn't call themselves SPF doctors. That's me. I call them SPF doctors. They're neurologists from the Massachusetts General Hospital, and there was another neurologist from Northwestern University in Illinois. I brought them to the neuro, all trying to show off what CBIG does. Remember my wow, holy jumping feeling I had in 2022 when I met this guy? Well, I want them to have that. It's all nice and dandy for me to do blah blah, but it's another kettle of fish for them to come see it with their own eyes. So on December 4th, there was a PLS. PLS stands for primary lateral sclerosis. It's another really ultra rare neurological disease. The SPF doctors were speaking at that engagement. PLS in Montreal, December 4th. And then there was the same doctors were speaking at an MND, motor neuron disease, MND conference in Montreal. So I said, okay, there's a gap on December 5th. So CBIG team and I, we crafted up an agenda. Patient on mission, but on a train again. And I came to Montreal and I went to the PLS conference and welcomed the doctors in person and brought them over to the neuro just to see what I found. That was one thing, just to expose everybody to their capabilities. Then on December 12th, we did a SPF Talks. SPF Talks is kind of, well, it is an ad hoc support group meeting. Any kind of subject, different times, it's just ad hoc, but it's always whatever subject works for patients, whatever will help them in their daily lives. So we did one of those on December 12th, exposing talking all about the white matter rounds that Dr. Lapiana from the neuro and her superstar lab assistant Juan Bedar, they work on this, on white matter rounds. So we brought that over to them. So brought that over to SPF Talks. When live at the meeting, there was almost 100 patients. But the meeting is also recorded and it's on YouTube. And last I checked, there was 428 views on that thing. So I was able to expose CBIG and their capabilities and Dr. Lapiana and their white matter rounds to 500 patients. Big deal. Another time I did that, a meeting on June, the other J month, January 9th, I exposed CBIG to CPATH. CPATH being an American foundation, they're working on rare diseases, they have a rare disease accelerator. I know we have stuff that can help them. Tried to figure out how to connect the dots there. Then closer to home, I did the same thing. This was on February 21st, February being Rare Disease Month. We did a webinar with ARQMO, ARQMO being the Regroupement Québécois des Maladies Orphelines, at home here, so it's the Quebec Coalition for Rare Diseases. It was called a Zebinar because, well, February is Rare Disease Month. You equivalent rare diseases with zebras. Play on words between zebra and webinar, it's a Zebinar. But we toyed with an idea there that's very interesting to me. Every time a patient of a rare disease gets a diagnosis of rare disease, it would be good to register them in research, and I fully agree. Now that I've had time to think about it, I love that idea because, well, registering in research maybe doctors don't have all the answers. Yeah, but registering them in research using open science, it's a way forward. That's what patients want, a way forward, just to do something. That's what See Big is. That's what Rockstar is. He's an opportunity. I love that idea. All right, I'm gonna wrap it up. I have a lot of fires in going on. I just don't have anything. All these examples, the 4 dates I just showed, they have a specific start date and a specific end. So I could present them with a nice bow for you. So, but I have a lot of things going on, some in Brazil, some in Quebec, some in Austria, some in the rest of Canada, and some in the States. It's just open science makes the connections they make. It takes the basic information from patients. This all starts with the basic information from patients, and it makes it explode. This is all on a journey to get care and cure for patients. It all starts with patients. It all ends with patients. Thank you.